Welcome to the Concierge Care Health System podcast. The information shared in this podcast is for general informational purposes only and does not constitute medical advice. Listeners should be aware the content is not a substitute for professional medical advice, diagnosis, or treatment. Always consult with a physician or other qualified healthcare provider for medical questions.
Welcome to the Deep Dive. Today we're diving into a topic that has just profoundly reshaped human existence.
Yeah?
Vaccines.
It really has.
I mean, this isn't just a medical marvel.
Yeah.
It's one of the most transformative public health interventions in history. We're talking preventing an estimated 2 to 3 million deaths every single year.
Yeah. The scale is hard to even grasp.
Just imagine the sheer amount of suffering averted.
Yeah.
This tech has truly fundamentally altered our relationship with infectious diseases.
Indeed. And what's truly remarkable, I think, is seeing how this technology evolved, from these ancient empirical observations, almost like folklore,
Right.
to today's incredible precision molecular biology and even, AI-driven platforms. It's a journey that tells us so much about scientific progress, but also human ingenuity.
That's right. So our mission in this deep dive is really to unpack that incredible journey. We'll look at how far vaccine technology has come using some pivotal public health case studies.
Mm-hmm.
And then we'll confront some of the complex societal challenges that are maybe surprisingly really constraining its full potential now.
Yeah, that's a crucial piece.
You see, the future of vaccines, it depends just as much on overcoming these political barriers as it does on scientific innovation. So by the end of this deep dive, you'll hopefully have a shortcut to being well-informed. We'll give you surprising facts, crucial insights into not just the science, but the very human story behind it all.
Absolutely. Helping you understand why its future depends on both science and society.
Okay, so let's start at the very beginning. Long before microscopes, before we even really understood germs, ancient cultures were already developing ways to protect against smallpox.
Right, this practice called variolation, or sometimes inoculation.
Yeah, and its earliest roots seem to be in China, by at least the 15th century, maybe even earlier, some say 200 BCE.
Possibly that early, yeah. The first really solid written description we have dates to 1549 in China. And what's truly fascinating is how this empirical knowledge, this, almost folk medicine, traveled across continents.
How did it actually work?
Well, the procedure involved deliberately introducing material from the pustules, the sores, of someone who had a mild case of smallpox into a healthy person.
Right.
Typically by scratching it into the skin or sometimes by inhaling a dried scab powder. Sounds pretty grim, I know.
A little bit, yeah.
From China, it made its way to the Ottoman Empire by the 1500s. And it was there that Lady Mary Wortley Montagu.
Ah, yes, the ambassador's wife.
Exactly, the wife of the British ambassador.
Right.
She saw how well it worked and played a really key role in introducing it to Europe in 1721.
And didn't it reach America around the same time, through a different route?
It did. Remarkably, around that same time, knowledge of variolation came to the American colonies through an enslaved West African man named Onesimus. He shared the practice with Cotton Mather in Boston, right in the middle of a terrible smallpox epidemic in 1721. So you have these parallel introductions happening.
So while it sounds like a clever idea for the time, albeit risky, what were the actual risks involved?
Well, you're right to flag the risks. Despite it definitely reducing mortality compared to just getting smallpox naturally, variolation wasn't safe.
How so?
The infection they induce could sometimes be severe, even fatal. And, maybe more importantly, someone who was inoculated could actually transmit the full-strength deadly disease to other people around them.
Ah, so you could start an outbreak just by trying to prevent one.
Precisely.
And yet it gained a really powerful advocate, Benjamin Franklin. Wasn't it after his own son?
Yes, tragically. His son died from smallpox in 1736. But Franklin, being Franklin, went beyond just personal tragedy or anecdote. In 1759, he published this analysis using quantitative data, some of the earliest public health data we know of.
What did it show?
It showed significantly lower death rates from variolation compared to getting smallpox naturally. This statistical evidence wasn't just, helpful. It was a pivotal moment. It's kind of the birth of evidence-based public health.
Wow, using data itself as a life-saving tool way back then.
Exactly, long before modern epidemiology. And it became accepted enough that General George Washington mandated it for all new recruits in the Continental Army during the Revolutionary War.
Really? To protect the army.
Yes, it protected his forces from the epidemics that absolutely ravaged other armies at the time. A strategic advantage, really.
So this naturally leads to the next step, right?
Yeah.
How did we move from this fairly risky variolation to something safer?
Right. That decisive leap came with Edward Jenner, an English physician. While variolation used the smallpox virus itself, Jenner's innovation, well, it came from local folklore.
The stories about milkmaids.
Exactly. The observation that milkmaids who got cowpox, a much milder disease, seemed to be protected from smallpox. So Jenner's idea was to use this milder related virus cowpox to confer immunity.
And he did that famous experiment in 1796.
Yes, May 1796, he took fluid from a cowpox sore on a milkmaid, Sarah Nelms, and inoculated an 8-year-old boy, James Phipps.
What happened to the boy?
Phipps got a mild fever, a bit of a reaction where he was inoculated, but recovered quickly. Then, 2 months later, Jenner did something pretty bold.
He exposed him to smallpox.
He did! He exposed Phipps to material from a fresh smallpox lesion, and Phipps remained perfectly healthy, immune.
Wow.
And Jenner coined the term vaccination from vaca, the Latin word for cow, to distinguish his method from variolation.
So it wasn't just the idea, it was the proof.
Exactly. Jenner's real contribution wasn't just noticing the milkmaid connection, it was the scientific rigor. He meticulously documented his cases, carefully distinguishing real cowpox from other sores that didn't protect. He published his findings in 1798.
That systematic approach.
Yes, that's why he's called the father of vaccines. And his method completely eliminated the risk of causing severe smallpox or spreading the deadly disease.
Now, I remember reading there's some initial skepticism. Like people thinking you would turn them into cows.
Oh, yes, there were cartoons and rumors like that. It sounds wild today, but, it was a new, strange idea.
I suppose so. But it caught on.
It did, relatively quickly. It gained widespread recognition, got endorsed by big names like Napoleon Bonaparte and Thomas Jefferson. And the first U.S. vaccinations were done by Dr. Benjamin Waterhouse in Boston, starting in 1800.
Okay, so we have Jenner establishing the practice. How did these ideas then get scaled up, become more, scientific?
Well, if Jenner established vaccination, it was really Louis Pasteur, the French chemist and microbiologist who propelled it into the modern scientific era. This is the latter half of the 19th century.
Moving from observation to the lab bench.
Precisely. His work fundamentally shifted the field to rigorous laboratory science, and it helped firmly establish the germ theory of disease.
And his key idea was attenuation, right? Weakening the germ.
Exactly. The principle of attenuation, weakening a pathogen so it can't cause serious disease, but it still triggers a protective immune response. This became foundational for so many 20th century vaccines.
Did he have early successes with that?
He did. His first big one was in 1872 with a vaccine for fowl cholera in chickens. He found that old cultures of the bacteria lost their virulence. When he injected these aged cultures, the chickens got mildly ill but survived, and crucially, they were then protected against the fully virulent bacteria.
Ah, proof of concept.
Yes. But he achieved even greater public fame with his 1885 rabies vaccine.
Rabies, wow. That was a death sentence then.
Absolutely terrifying and universally fatal. Pasteur developed a method of weakening the rabies virus by drying infected rabbit nerve tissue. And despite not actually being a medical doctor,
Right, he's a chemist.
he dramatically succeeded with his post-exposure treatment for young Joseph Meister, a boy who'd been badly mauled by a rabid dog. Meister survived. It was a huge public demonstration, got global attention.
That must have been transformative for public perception.
Completely. Connecting this to the bigger picture, Pasteur's work, alongside others like Robert Koch, solidified the germ theory. It really opened what we call the golden age of vaccines.
Leading to vaccines for
Diphtheria, tetanus, pertussis, polio, measles, mumps, rubella. Many of the vaccines we still rely on today have roots in this era.
And it seems like a pattern emerges. Scientific breakthrough needs to be paired with building public trust.
Always. Just like Franklin used statistics, Jenner used careful documentation, and Pasteur had these very public successes. That trust-building part has always been crucial for adoption.
Okay, so that brings us closer to the modern era. You mentioned this theme of progressive refinement, from using the whole germ to just parts of it, or even just its blueprint.
Exactly. That's the central theme of modern vaccine development. Moving from whole pathogen to pathogen part to pathogen blueprint. It's a shift from biological manufacturing towards more synthetic, precise, and faster approaches.
Let's unpack those. The earliest modern ones, based on Pasteur's work, used whole pathogens.
Right. Two main types there. First, Live attenuated vaccines. These contain a weakened, live version of the virus or bacterium.
Like the MMR vaccine.
Exactly. MMR for measles, mumps, rubella, or the Sabin oral polio vaccine. They replicate inside you, mimicking a natural infection, which usually gives you really strong, often lifelong immunity with just 1 or 2 doses.
What's the downside?
Well, because they are live, even though weakened, they generally can't be given to people with severely weakened immune systems.
Yeah.
There's also a very, very rare risk the pathogen could revert to a more dangerous form, and they often need careful cold storage, the cold chain.
Okay. And the other type, using whole pathogens.
Those are inactivated or killed vaccines. Here, the pathogens are completely killed, usually with chemicals or heat.
Like the Salk polio vaccine. Or flu shots.
Yes, the Salk inactivated polio vaccine, hepatitis A vaccine, rabies vaccine, most standard flu shots. Because the pathogen is dead, it can't replicate or cause disease. This makes them extremely safe, suitable for pretty much everyone, including immunocompromised people.
But what's the trade-off there, if they're so safe?
The trade-off is they're usually less immunogenic. They don't provoke as strong an immune response as live vaccines.
Meaning you need more shots.
Exactly. You almost always need multiple initial doses, often periodic boosters later on. And they frequently include adjuvants.
Adjuvants, what are those?
They're substances added to the vaccine to help stimulate a stronger immune reaction, basically making the vaccine work better.
Okay, so that's whole pathogens. Then we move to using just specific parts.
Right. That's the next level of refinement. We have toxoid vaccines and subunit or protein vaccines.
Toxoids. Like for tetanus.
Precisely. Diphtheria and tetanus vaccines are toxoid vaccines. They don't target the bacteria themselves, but the harmful toxins the bacteria produce. The vaccine contains an inactivated version of the toxin called a toxoid.
So your body learns to fight the toxin, not the bug itself.
Exactly. It teaches your immune system to make antibodies that neutralize the toxin, preventing the disease's worst effects. They give a strong antibody response, but they don't stop you from being colonized by the bacteria.
Okay. And subunit or protein vaccines?
These go even further. They use only specific, purified pieces of the pathogen, like certain proteins or sugars, polysaccharides, from its surface that are known to trigger a good immune response.
So even safer.
Very safe, yes, because they contain no live components at all. They can be used in almost everyone. Think of the hepatitis B vaccine or the human papillomavirus, HPV vaccine.
There are challenges of these.
Well, figuring out which specific pieces, which antigens are the best ones to use can be complex. And like inactivated vaccines, they often need adjuvants and multiple doses to get strong, lasting immunity.
You mentioned HPV. Aren't there variations like conjugate vaccines or VLPs?
Good point. Yes, within subunits there are clever variations. Conjugate vaccines were a big breakthrough for protecting babies. They link a polysaccharide sugar antigen, which infants' immune systems don't respond well to on its own, to a carrier protein that does trigger a strong response, like the hive vaccine.
Okay, and VLPs.
Virus-like particles, or VLPs. These are amazing. They're engineered protein shells that look like a virus to the immune system. They mimic its shape, but they contain absolutely no genetic material, so they can't cause infection but are highly immunogenic. The HPV vaccine is a prime example of VLP technology.
So the progression is clear. Whole germs, then key parts.
Yeah.
But the evolution didn't stop there, did it? We had this genetic revolution.
Absolutely. The late 20th and especially the 21st century brought platforms that are fundamentally different. They don't deliver the antigen itself.
They deliver the instructions.
Exactly. They provide the genetic instructions, the blueprint for the recipient's own cells to manufacture the antigen. This offers unprecedented speed and flexibility in development and manufacturing, which we saw very clearly during the COVID-19 pandemic.
Okay, tell us about these genetic blueprint platforms. Viral vectors first.
Right. Viral vector technology. This uses a harmless modified virus, often an adenovirus like the ones that cause common colds, but engineered so it can't replicate as a delivery vehicle.
A vector.
Yes, a vector. It carries the genetic code for the target antigen, say the SARS-CoV-2 spike protein, into your cells. Your cell machinery then reads that code and produces the spike protein.
And the immune system reacts to that protein.
Correct. Because the antigen is made inside the cell, these vaccines tend to generate a very strong T-cell response, which is important for clearing infected cells, in addition to a good antibody response. Examples are the Johnson & Johnson Janssen and AstraZeneca COVID-19 vaccines, and also a very effective Ebola vaccine.
Any drawbacks to vectors?
The main potential challenge is pre-existing immunity. If you've already been infected by the specific type of adenovirus used as the vector, your immune system might attack the vector itself before it can deliver its payload effectively, potentially reducing the vaccine's efficacy.
Okay. And then the technology that everyone knows now, messenger RNA, mRNA vaccines, these seem to appear overnight, but the research goes way back, doesn't it?
Decades back. Absolutely. The concept is really elegant. The vaccine delivers a synthetic strand of messenger RNA protected inside a tiny fat bubble called a lipid nanoparticle, or LNP. This mRNA provides the instructions for your own cells to make a specific antigen, like the spike protein again.
Your cells become temporary vaccine factories.
In a way, yes. They make the antigen, display it, and that triggers the immune response. And it's absolutely vital to stress, as we always do. The mRNA works in the cell's cytoplasm. It never enters the nucleus where your DNA is stored, and it doesn't alter your DNA in any way. It's a temporary message that gets broken down quickly.
And the breakthroughs that made it possible.
Key moments include Catalin Carrico and Drew Weissman's work around 2005. They figured out how to modify the mRNA chemically so it could slip past the body's initial inflammatory sensors. And Peter Cullis's group made huge strides in developing the lipid nanoparticle delivery systems, getting the fragile mRNA safely into the cells. Those were crucial.
Okay. Finally, in this genetic category, we have DNA vaccines. How do they fit in? Are they similar to mRNA?
Similar principle, different molecule. DNA vaccines use a circular piece of DNA called a plasmid, which contains the gene for the target antigen.
So DNA instead of RNA instructions.
Right. When injected, some of your cells take up this DNA plasmid, but then the DNA has to travel all the way into the cell's nucleus to be transcribed into mRNA. Then that mRNA goes out to the cytoplasm to be translated into the antigen protein.
So an extra step compared to mRNA vaccines.
Exactly. They are very stable, easy to produce, and the plasmid DNA itself can act as a kind of natural adjuvant, boosting the immune response. But that step of getting the DNA into the nucleus efficiently in humans has been a major hurdle.
Meaning they don't work as well in people?
They tend to have lower immunogenicity in humans compared to what's seen in animal studies or compared to mRNA or vector vaccines. So, to date, no DNA vaccine has actually been licensed for human use. They are used successfully in veterinary medicine, though, like the West Nile virus vaccine for horses.
That's really fascinating. Understanding these different platforms, whole, parts, blueprint, really highlights the incredible ingenuity over the years, doesn't it? How far we've come.
It really does. Each step represents a leap in precision and understanding.
Okay, let's turn to some major case studies now. The ultimate proof of concept. The global eradication of smallpox. I mean, this was a horrific disease, right? 30% mortality rate, killed over 300 million people in the 20th century alone.
Just staggering numbers. The World Health Organization launched its intensified smallpox eradication program in 1967. Several factors made eradication feasible.
What were they?
First, a highly effective vaccine based on the vaccinia virus. Second, crucially, smallpox only infects humans. It has no animal reservoir where it could hide.
So if you stop it in humans, it's gone.
Exactly. And third, infected individuals are usually easy to identify because of the distinctive rash, making outbreaks visible and trackable.
And the strategy wasn't just mass vaccination everywhere.
Not entirely. While mass vaccination played a role initially, the strategy shifted, especially in the later stages, to surveillance and containment, or what became known as ring vaccination.
Ring vaccination, how did that work?
When a case was identified, teams would rush in, isolate the patient, and then vaccinate everyone who might have had contact with them, family, neighbors, coworkers, creating a protective ring of immunity around the case to stop the virus from spreading further.
That sounds much more targeted and efficient.
It was highly effective, and this was helped by some key tech innovations, too. A stable, freeze-dried version of the vaccine that didn't need constant refrigeration, and, maybe surprisingly, the simple but brilliant bifurcated needle.
The two-pronged needle.
Yes. It held just the right dose of vaccine between its prongs and allowed for rapid, easy skin puncture, the multiple puncture technique. Minimal training needed, very effective.
So when did the last case occur?
The last naturally occurring case was recorded on October 26, 1977 in Somalia. The patient was a hospital cook named Ali Mao Malin.
Wow. Just one person, the end of a scourge.
And after 2 years of intense global surveillance with no new cases found, the WHO officially declared smallpox globally eradicated on May 8, 1980. A monumental achievement.
Truly. Now, polio. That's a more complex story, isn't it? Still ongoing.
Yes, poliomyelitis, the crippling viral disease that caused so much fear, especially in the mid-20th century. The fight against it involved two key vaccines.
Right, first there was Dr. Jonas Salk's vaccine in 1955. That was
The inactivated polio vaccine, or IPV. It uses killed poliovirus given by injection. It proved very safe and highly effective at preventing the paralytic form of the disease.
And then came Dr. Albert Sabin's vaccine in 1961.
Yes, the oral polio vaccine, or OPV. This one is live attenuated weakened live virus given as simple drops in the mouth.
Why develop a second vaccine? What were OPV's advantages?
OPV had several big advantages, especially for large-scale global campaigns. It was less expensive to make, much easier to administer, no needles, no highly trained vaccinators needed, you could use volunteers, and crucially, it induced superior intestinal immunity.
Intestinal immunity, why does that matter?
Because polio spreads through the fecal-oral route, OPV was better at stopping the virus from replicating in the gut and being shed in feces, which helped break the chains of transmission in communities. This made OPV the workhorse vaccine for the Global Polio Eradication Initiative, launched in 1988. It's responsible for reducing wild poliovirus by over 99%, an incredible success.
But, and there's a but, isn't there? The very nature of OTV being live created a unique challenge.
Indeed, a really paradoxical challenge. It's called circulating vaccine-derived poliovirus, or CVDPV.
Vaccine-derived polio, how does that happen?
Because OPV contains live, weakened viruses, they replicate in the gut and can be shed in the stool of vaccinated individuals for a few weeks. In communities with very low overall vaccination coverage, these shed vaccine viruses can start to circulate among unvaccinated people.
And they can change.
Yes. Over time, as they circulate person to person, these vaccine viruses can accumulate mutations. Rarely, they can mutate enough to regain their virulence and the ability to cause paralysis, just like the original wild poliovirus.
So the vaccine itself can, in rare circumstances, lead to polio outbreaks.
In areas with chronically low immunization rates, yes. And today, paradoxically, these CVDPV cases actually outnumber the cases caused by the remaining wild poliovirus strains.
Wow. So what's the endgame strategy then?
It's complex. It involves a phased withdrawal of OPV types, starting with the type 2 component, which was responsible for most CVDPV cases, and eventually moving towards the exclusive use of the Salk IPV globally. IPV, being killed virus, carries zero risk of causing vaccine-derived polio, but it's a huge logistical and financial challenge.
A really complicated final chapter. Now let's talk measles. You said it illustrates herd immunity perfectly.
Yes, and the fragility of it. Measles is incredibly contagious, one of the most infectious viruses known. Before the vaccine came along in 1963, it caused millions of deaths globally every year.
And the vaccine is very effective.
Extremely. The MMR vaccine 2 doses give about 97% lifelong protection. It's a fantastic tool.
And its success led to the U.S. declaring measles eliminated back in 2000. That relied on herd immunity, right? Explain that concept again.
Herd immunity, or community immunity, is simple in principle. When a high enough percentage of a population is immune to a disease, either through vaccination or prior infection, it creates a protective barrier. The virus can't find enough susceptible hosts to sustain transmission. This protects everyone, but especially the most vulnerable, who maybe can't be vaccinated themselves, like infants or immunocompromised people.
And for measles, because it's so contagious, you need really high coverage.
Exceptionally high. The herd immunity threshold for measles is estimated to be between 93% and 95% vaccination coverage with 2 doses. You need almost everyone protected to keep it out.
So if it works so well, and we eliminated it, why are we seeing measles outbreaks again in places like the U.S. and Europe?
This reveals what some call the paradox of success.
Paradox of success.
The very effectiveness of vaccines makes the diseases they prevent seem like ancient history. People forget how bad measles, polio, or diphtheria really were. When you don't see the disease, the perceived risk from the disease plummets.
And the focus shifts to vaccine side effects.
Exactly. Concerns about vaccine side effects, however rare or even completely unfounded, can become magnified in people's minds when the disease itself feels distant or non-threatening. This can fuel vaccine hesitancy,
leading to drops in vaccination rates.
Yes, and because the herd immunity threshold for measles is so high, even relatively small drops in vaccination coverage in a community can create enough susceptible individuals for the virus to gain a foothold if it's introduced, perhaps by an unvaccinated traveler. These gaps breach the herd immunity wall,
allowing forgotten diseases to reemerge.
Precisely. It's a stark reminder that elimination is not eradication. Complacency, fueled by success, combined with misinformation, can undo decades of public health progress.
Okay, let's pivot to that social dimension more directly. Vaccine hesitancy and misinformation. It feels very modern, very internet-driven, but you're saying opposition is much older.
Oh yes, nearly as old as vaccination itself. The first organized anti-vaccination movements sprang up in 19th century England.
In response to the smallpox vaccine.
Directly in response to government mandates for the smallpox vaccine, like the Vaccination Act of 1853, which made it compulsory for infants.
What were their objections back then?
They were varied. Some had genuine sanitary concerns about the arm-to-arm method used early on. Others had religious objections. Injecting material from a cow seemed unchristian or unnatural to some. And there was a strong undercurrent of distrust in the medical establishment and government authority, a feeling of infringement on personal liberty.
So organized resistance.
Very organized. Groups like the Anti-Vaccination League formed. They held rallies, published pamphlets. There was a massive demonstration in Leicester in 1885. Estimates range from 80,000 to 100,000 protesters marching against compulsory vaccination.
Wow. And in the U.S.?
Similar debates happened. It culminated in a landmark Supreme Court case in 1905, Jacobson v. Massachusetts. The court upheld the state's authority to mandate smallpox vaccination for the public good, establishing a legal precedent. But that fundamental tension between individual rights and collective health has obviously never gone away.
So hesitancy faded a bit as diseases declined, but then reemerged later.
It did. There's a notable controversy in the UK in the 1970s around the DTP vaccine, diphtheria, tetanus, pertussis, linking it to neurological issues. Vaccination rates dropped, and there was a major whooping cough epidemic as a result.
And then came the MMR controversy.
Yes, the most damaging modern example. In 1998, British physician Andrew Wakefield published a study in the prestigious medical journal The Lancet, suggesting a link between the MMR vaccine and autism.
That study's been thoroughly debunked.
Completely. It was found to be fraudulent based on manipulated data involving only 12 children. The paper was retracted by The Lancet. Multiple large-scale epidemiological studies found absolutely no link, and Wakefield was stripped of his medical license.
But the damage was done.
Immense damage. The claim was sensationalized by media, amplified by celebrities, and it took root online. It persists to this day despite overwhelming scientific evidence against it, significantly damaging public trust and directly contributing to falling MMR vaccination rates and the measles outbreaks we just discussed.
So why is vaccine hesitancy still such a major issue today? You mentioned the WHO calls it a top global health threat.
It's incredibly complex, truly multifaceted. There's rarely just one single reason why someone might be hesitant.
Can we break down some of the drivers?
Sure. At a personal or psychological level, you have things like a preference for natural immunity or cognitive biases. People tend to overestimate small risks like rare vaccine side effects while underestimating the risk of the disease if they haven't seen it. Negative past experiences with healthcare can also play a role.
Okay, what about social factors?
Social and cultural factors are huge. We live in echo chambers, both online and offline, where misinformation can circulate unchecked among like-minded people. Certain religious or cultural beliefs might raise concerns about vaccine ingredients like gelatin. And crucially, there's deep-seated distrust in medical and government institutions.
Especially among certain communities.
Yes, particularly among marginalized communities with histories of mistreatment by the medical system or government. Think of the horrific Tuskegee syphilis study in the U.S., where treatment was deliberately withheld from black men, or more recently, reports of the CIA using a fake vaccination program in Pakistan during the hunt for Osama bin Laden. These aren't abstract issues. They create a real, understandable legacy of distrust that makes people wary.
That's a really important context. And systemic factors.
Environmental or systemic factors also matter. Socioeconomic status can influence access to reliable information and healthcare itself. And in some places, like the U.S. currently, vaccination has become intensely politicized. Resistance to mandates gets framed as a defense of personal freedom, detached from the public health implications.
So given all that complexity, how do you even begin to address hesitancy? Confronting people doesn't work, right?
Generally, no. Confrontational tactics, just throwing facts at people, often backfire. They can make people defensive and double down on their beliefs. Effective strategies need empathy, understanding the specific concerns, and tailored communication.
Who is best placed to do that?
Healthcare providers are consistently ranked as the most trusted source of information on vaccines. Empowering doctors, nurses, pharmacists to have these conversations is key.
Using specific techniques.
Yes, things like motivational interviewing. It's a patient-centered approach. Instead of lecturing, you use open-ended questions, listen reflectively, try to understand their perspective, and help them explore their own reasons for or against vaccination. It's about guiding them to their own motivation, not forcing yours on them.
And visual aids?
Clear risk communication tools can help too, like simple icon arrays that visually show the relative risks of the disease versus the vaccine side effects, making abstract numbers more concrete.
What about tackling misinformation more broadly at a public health level?
There are different strategies. Debunking directly correcting false claims is necessary, but it can sometimes inadvertently reinforce the myth just by repeating it. So there's growing interest in pre-bunking, also called inoculation theory.
Inoculating against bad information.
Kind of. The idea is to warn people in advance about the manipulative tactics used to spread misinformation. Things like using fake experts, cherry-picking data, appealing to emotion, conspiracy theories. If people recognize the tactics, they're less likely to fall for the misinformation when they encounter it.
Interesting. And monitoring the landscape.
Yes, using social listening systems to track emerging rumors and false narratives online, and then engaging communities through trusted local figures, religious leaders, community organizers to provide accurate information from sources people already trust.
Okay, let's widen the lens again to the global dimension. The COVID-19 pandemic didn't just highlight hesitancy, it exposed massive global inequities in access, didn't it?
In the starkest possible terms. It laid bare the profound gap between wealthy nations and low-income nations when it comes to accessing life-saving medicines.
Wasn't there an initiative, COVAX, designed to prevent that?
Yes, COVAX was launched led by the WHO, Gavi, the Vaccine Alliance, and CEPI, Coalition for Epidemic Preparedness Innovations. It was an ambitious, unprecedented effort to try and guarantee fair and equitable access to COVID-19 vaccines for all countries, regardless of their ability to pay.
That didn't quite work out as planned.
It was systematically undermined, primarily by vaccine nationalism.
Vaccine nationalism, meaning countries looking out for themselves first.
Exactly. While COVAX was trying to pool resources and negotiate for doses for the whole world, wealthy nations were making massive side deals directly with the pharmaceutical companies, pre-ordering enormous quantities of vaccines, often enough to cover their own populations multiple times over.
Hoarding the supply.
Essentially, yes. This hoarding created artificial scarcity in the global market, drove up prices, and severely weakened COVAX's negotiating power. The result was, by early 2021, high-income countries, with only about 14% of the world's population, had secured over half of the most promising vaccine supply. Meanwhile, vaccination rates in many low-income countries were stuck near zero. It was a catastrophic market failure and a moral failure.
Were there other barriers for lower-income countries besides just not getting the doses?
Oh, absolutely. Even when doses eventually started to arrive, low- and middle-income countries, LMICs, faced huge hurdles. Lack of robust supply chains and logistics, especially for vaccines requiring ultra-cold storage. Insufficient funding for the last-mile delivery paying vaccinators, transport, community outreach, chronic shortages of trained healthcare workers to administer the shots, and complex regulatory hurdles, as many national authorities lack the capacity to rapidly review and approve brand-new vaccines.
So, connecting this back, why is this inequity not just unfair, but bad for everyone?
Because, as we learned painfully, in a pandemic, no one is safe until everyone is safe. Failure to ensure equitable access doesn't just prolong the suffering in poorer countries. It allows the virus to continue circulating widely, increasing the risk of new, potentially more dangerous variants, emerging variants that could evade existing vaccines and threaten everyone, everywhere.
So it's a global public health imperative.
Absolutely. Plus, it reinforces historical narratives of exploitation and neglect, further eroding trust in global health institutions and making populations potentially more susceptible to misinformation and hesitancy down the line. It's all interconnected.
Okay, let's shift gears and look towards the horizon. The future of vaccinology sounds pretty mind-bending. Beyond the mRNA we know, what's next? Self-amplifying RNA.
Yes, self-amplifying RNA, or SARNA. This is a really exciting next step. It's not just standard mRNA. It includes an extra piece of genetic code, usually borrowed from a virus, that acts as a replicase enzyme.
Replicase, meaning it makes copies.
Exactly. Once the SARNA gets inside the cell, the replicase machinery makes multiple copies of the antigen-encoding RNA strand.
So the cell produces way more antigen from the initial dose.
Precisely, and that has a profound implication. dose-sparing.
Meaning you need less vaccine.
Much smaller doses might be needed to achieve the same or even greater level of antigen production and immune response compared to conventional mRNA.
Which means?
Cheaper manufacturing, potentially fewer side effects because you're injecting less material, and critically, getting many more doses out of the same amount of raw materials. That's huge for pandemic response or for making vaccines more affordable globally.
Is this real yet? Or just theoretical?
It's becoming real. The first mRNA vaccine for COVID-19 actually received regulatory approval in Japan in late 2023. There are still challenges. The mRNA molecule is larger and more complex than mRNA. It might cause slightly higher, but temporary side effects initially, reactogenicity. And there's a theoretical, though probably very low, risk of recombination events. But the potential is enormous.
Wow. Okay, what about the Holy Grail? Yeah. A universal flu vaccine, no more guessing games each year.
That's the dream. The quest for a universal influenza vaccine that would protect against most or all strains season after season, maybe even for life.
What makes that so hard?
The main challenge is the flu virus's constant shape-shifting, a process called antigenic drift. The proteins on its surface, especially hemagglutinin or HA, which current vaccines target, change rapidly from year to year. That's why the vaccine has to be reformulated annually based on predictions of which strains will circulate.
So how would a universal vaccine work?
The strategy is to redirect the immune response away from those rapidly changing parts, like the head of the HA protein, towards parts of the virus that are conserved, meaning they change very little across different strains and subtypes.
Like what parts?
A major focus is the stalk region of the HA protein. It's much more stable than the head. Other targets include another surface protein called M2E, or even internal viral proteins that could stimulate protective T-cell responses.
And the new platforms are helping here?
Absolutely. Platforms like mRNA and advanced nanoparticle vaccines are perfectly suited for presenting these conserved antigens in ways that elicit broad, potent immune responses. There are literally over 200 candidates in development, dozens in clinical trials. We're closer than we've ever been to cracking this.
That would be incredible. Now, maybe the most revolutionary idea, vaccinating against cancer. Is that really possible?
It's a rapidly advancing frontier. Therapeutic cancer vaccines. These aren't preventative like childhood vaccines. They're designed to treat existing cancer by training the patient's own immune system to recognize and kill tumor cells.
How does that work? It sounds like personalized medicine.
It is truly personalized medicine at its most sophisticated. The core idea is to target neoantigens.
Neoantigens, new antigen.
Yes. These are unique, abnormal proteins that arise specifically because of the random genetic mutations that occur within a patient's tumor cells. They are present on the cancer cells, but not on the patient's healthy cells.
So they act like flags identifying the cancer cells as foreign.
Exactly. The process usually involves taking a sample of the patient's tumor, sequencing its DNA and RNA to identify these unique neoantigen mutations, using computational algorithms, AI, actually to predict which ones are most likely to trigger a strong immune response.
And then making a custom vaccine.
And then designing a custom mRNA vaccine that encodes a selection of that patient's specific neoantigens. When this personalized vaccine is administered, the patient's cells produce these neoantigens, effectively showing them to the immune system and training T cells to find and destroy any cancer cell displaying those same markers.
That's incredible. Is it working in trials?
We're seeing really promising early results, especially when these personalized mRNA vaccines are combined with other immunotherapy drugs called immune checkpoint inhibitors. Trials in high-risk melanoma patients have shown significantly reduced rates of cancer recurrence or death compared to the standard therapy alone.
Wow. Any other cancers?
There's exciting work happening in pancreatic cancer, colorectal cancer, lung cancer. It's still early days for many applications, but the potential is immense. Researchers are also exploring off-the-shelf cancer vaccines targeting common tumor antigens, which wouldn't be personalized but could potentially work for broader groups of patients.
It feels like the pace of all this innovation is accelerating dramatically. What role is artificial intelligence, AI, and machine learning playing in this?
A massive and growing role. AI is supercharging vaccine development at almost every stage.
Like finding those neoantigens?
Yes, for antigen discovery and design. AI algorithms can sift through vast amounts of genomic data to predict which parts of a pathogen or which neoantigens on a tumor will make the best vaccine targets. AI is even being used to design entirely new protein structures from scratch that could serve as optimal antigens.
What about making the vaccines better?
AI helps in optimizing formulations and delivery. Machine learning models can predict the stability of different mRNA sequences or lipid nanoparticle formulations, potentially helping design vaccines that are more stable at higher temperatures, reducing the need for ultra-cold chains.
And speeding up testing.
Absolutely. In clinical trials, AI models might help predict vaccine efficacy and safety even before human testing begins, potentially reducing reliance on animal models. During trials, AI can analyze incoming data in real time to help optimize dosing strategies or identify subgroups who respond differently.
So it's like the genetic platforms are the hardware and AI is the software.
That's a great analogy. The genetic platforms give us the ability to write biological code, and AML provides the tools to design the optimal code much faster and more effectively than ever before. This synergy is really driving the vision of programmable medicine, rapidly designing tailored vaccines for future pandemics, personalized cancer therapies, maybe even that universal flu shot.
What an incredible journey we've covered today, really decoding the world of vaccines.
Yeah.
From those ancient roots of variolation spreading across continents.
Yeah, that empirical knowledge.
To the scientific revolutions sparked by Jenner and Pasteur, really setting the stage. And now, these cutting-edge AI-driven genetic platforms, we've seen this amazing march towards more precision, better safety, incredible speed.
Culminating in triumphs like smallpox eradication, which still feels miraculous.
It really does. But this brings us right back to that central tension we identified. While the science is accelerating at this blistering pace.
Yeah, the innovation is stunning.
The main roadblocks, the constraints on vaccines actually saving lives everywhere, seem increasingly social, political, logistical.
Exactly. We've talked about the deep historical roots of vaccine hesitancy, how it's amplified today by misinformation spreading online, and then the stark global inequities that COVID just threw into sharp relief, undermining efforts like COVAX.
And it's crucial to see how those things connect, right? Inequity and hesitancy aren't totally separate issues.
Not at all. They can form a toxic feedback loop. When people feel neglected or exploited by the global health system, or have historical reasons for distrust, that provides fertile ground for misinformation to take root and flourish. Building trust is fundamental to addressing both.
So the future of this life-saving technology, scientifically, it looks incredibly bright. SARNA, universal vaccines, cancer therapies.
The scientific horizon is amazing, yes, but its full potential actually reaching people and having the impact it could. That really depends on a parallel investment in what you might call the software of public health. Meaning building health systems that are truly equitable and accessible to everyone, developing communication strategies that are empathetic, that meet people where they are, engaging communities through trusted local voices, and frankly, fostering a global political commitment to solidarity over narrow self-interest.
So as you, our listener, reflect on this deep dive, maybe the final thought is this. The science is creating marvels, but what role can you play in your community, in your conversations, in fostering the trust and understanding needed to ensure these marvels actually reach everyone who needs them?
That's the critical question for all of us, I think.
Concierge Care, providing world-class, personalized care for patients and members. Taking a proactive approach by offering its free digital health clinic for anyone in need of these services. Reach out to us via our website, www.health.conciergecare.net, or give us a phone call at 786-809-2345.